Good morning, everybody, and thank you very much for joining our SPHN webinar. We are very happy to present you today a guest speaker and PI of a project funded by the SPHN sister initiative, PHRT, the Personalized Health and Related Technologies of the ETH domain. The project is a PHRT technology transfer project in the imaging field entitled A Translational Platform for New Theragnostics, with the aim of improving the availability of radionuclides and radiotracers for clinical studies by establishing a respective radiopharmacy platform. And we are, of course, very keen on getting to know the project in more detail and to hear what has been achieved in the realm of this project since its start in 2017. And with this, I would like to warmly welcome our speaker of today, Professor Roger Schibli. Roger is a professor at the Department of Chemistry and Applied Biosciences at the ETH in Zurich, and also a laboratory head at the Paul Scherrer Institute, which is part of the ETH domain. In addition, Roger is leading the Center for Radiopharmaceutical Sciences. This is a joint endeavor between the ETH Zurich, the Paul Scherrer Institute, and the University Hospital in Zurich. Roger is a chemist by training and was appointed full professor at ETH in 2010. His research is directed towards targeted tumor diagnosis and therapy using radio-labeled compounds with a strong focus on the development of new radionuclides, identification of novel targeting molecules, and their biological pharmacological characterization. And of course, to bring these new radiopharmaceutical drug candidates into clinical trials to close the loop to the patient. So I'm very grateful to give the floor to you now, Roger. Thank you very much for taking the time and being with us today. The screen is yours.
Okay, thank you very much. I hope everybody can hear me. So it's really a very nice introduction, and I think it covers already a few aspects which I want to point out. It's really a joint endeavor between the ETH in Zurich and also the Paul Scherrer Institute. So both institutions are involved in this project. I'm very grateful that this project has been funded in 2017, and I'm going to give you a few examples of what we have achieved in the meantime. So maybe just for you as a general introduction, so what are radiopharmaceuticals? Radiopharmaceuticals are medications, medical products, which comprise a radioactive isotope in their structure, and by the decay properties of those isotopes, these radiopharmaceuticals can be used for diagnosis and also for therapy. So they also comprise, based on that, on two components. So it's the radionuclide, but also a biological entity that could be a small molecule, a peptide, or a protein, which is basically giving the targeting or giving the capacity for this radiopharmaceutical to go to the sites where they should go. They have actually, radiopharmaceutical have a unique opportunity because of the physical decay properties of the isotopes which are used. We can use for diagnosis isotopes which emit gamma particles or gamma radiation, as well as beta plus particles, which then lead to annihilation. So these are standard isotopes which are used in the clinics at the moment: fluor-18, gallium-68, technetium-99m. And on the other hand, we have also isotopes which are decaying by particle emission, like alpha particles and beta minus particles. And these isotopes can be used for therapy of cancer lesions, for example. Radiopharmaceuticals are applied systemically, so you inject them into a patient and they go to the sites where they should go by means of their properties, targeting capacity. So you might wonder, why is that something which we are bothering in academia, particularly when it comes to translational research, and how can we actually do translational research within, in academia from bench to bedside? That's another peculiarity of radiopharmaceuticals, because they really applied at minute quantities. So we're talking about nanograms to micrograms of substance, of cold material, of biological material you're going to inject. So that means we can adhere to the so-called microdosing principles. That means we do not expect any pharmacological effect from these radiopharmaceuticals when injected, but it's only the physical properties which are actually leading to a diagnosis or therapeutic application. So that's also something which has to be considered, which has, of course, implications into dosimetry for dosing and also for toxicity studies, because they do not need extensive chronic toxicity studies. So nuclear imaging, and basically what we produce are the tracers for which are used in nuclear medicine. So these radiopharmaceuticals are really important because nuclear medicine, is trying to characterize biological processes at the cellular and also the molecular level. And of course, through that, they also would contribute, or nuclear medicine contributes to the personalized treatment options. And I think that is also something one has to really emphasize. We are not looking at structures, we are looking at function of an organ, we're looking at function of a disease, and not really on the morphology. That's also a very important aspect because of this high sensitivity which we can achieve with this radio tracer because of the small quantity you apply. So the areas of application of radiopharmaceuticals are quite wide. So we have oncology, oncological applications, we have neurology, but also, of course, in cardiology, where functional information is important for patient management. Radiopharmaceuticals work with obviously radioactive material, and radioactive material, they are usually, these isotopes are short-lived. So we're talking about a few minutes up to a few days, half-life only. They need special equipment like accelerators. They need special equipment in terms of shielded hoods, which protect the personnel from radiation. So this is quite expensive equipment. Plus, we have to deal with short half-life. So the synthesis of a compound has to be accomplished, the very same day it's actually applied. And of course, nevertheless, these compounds are pharmaceutical, so they have to adhere to, certain regulations regarding safety, but also in terms of radiation safety. Okay, so this is really, that's in both sides. We have extensive costs, production, and also the application, and that leads very often to this. I call this radiopharmaceutical valley of death, where we have innovative tracers. We would like to have clinical imaging, but the costs and also the facilities which are producing such radiopharmaceuticals are not available, at least not in Switzerland, or not in Switzerland. So that was the starting point. So the aim was really to implement a Swiss-wide concept of automated radionuclide production and separation, and also incorporation in those for GMP, so for good manufacturing practice for radio tracers for clinical use. And we would like to support projects within the SPHN, but also in PHRT, and also beyond, that we can actually provide those with these tracers. So we had a starting point was actually already done. We had, of course, access to a cyclotron at the Paul Scherrer Institute and also the ETH. We had some hot cells already available. We also have preclinical imaging systems available. However, the GMP capacity at that time was still limited. When we started the project, we have GMP facilities at ETH, but also very recently also the Paul Scherrer Institute. So that was really the starting point. We also, as I mentioned before, we had mainly adhered to isotopes like fluorine-18 and carbon-11. But there's actually capacity and need for other radionuclides, which maybe have different decay properties more suitable. So the concept is indeed that we, have basically a clinical need. We have maybe academic partners. We also have industrial partners which, would like to establish a radio tracer for clinical use. But there is a lack of a platform which, facilitates it in Switzerland. And that's what we wanted to offer, and that's what we're currently offering. And of course, the output of that is that we have clinical studies ongoing. We generate IP for the academic environment, but also for industry. And of course, we push academic research. We have several projects in the pipeline and also applications, and I'm going to have a few examples shown here about Alzheimer tracer, ovarian cancer tracer for imaging of ovarian cancer, a therapeutic application for thyroid cancer, and also a few very recent examples of how to use the system for augmented reality for production, but also for future scanner development. So at the end, it's really important. We would like to serve the medical community. That's just a picture or two videos about the GMP facility at ETH Zurich. So you see this clean room facility. That's the system where how to produce such radiopharmaceuticals in an open situation. And of course, once you have the radioactivity, this is shielded now with lead shielded. And you can see here on the right side, you see the cyclotron facility at Hönggerberg, where we have actually installed a new facility and a new equipment. So what we did is, in order to increase the number of isotopes that could be used in nuclear medicine in Switzerland, besides fluorine and carbon-11, we had to establish a so-called solid target station. That means we can actually irradiate solid material, not only liquid and gas targets as we're currently doing. So that meant actually quite a substantial installation. So they had to drill holes in this system in our facility, establish a new hot cell here, and that's what you see here. That's this this solid target station where we can actually irradiate material to produce isotopes like scandium-44, copper, gallium-68, and zirconium, which also are useful in nuclear medicine. Parallel to that, we also established new automated equipment to produce radiopharmaceuticals independent from their isotope in use. So that's one of the projects, one of the first projects which were established. That was actually the university hospital asked, could we not produce a tau tracer for Taupathy, useful for the images and staging of Alzheimer patients. So they, from the literature, it was known that a company, Avid, they had produced and developed this compound, as seen on the right side, but it was not available in Switzerland for clinical studies. And there were nobody at the moment, at this point, available to produce it, and it was also not possible to import this radiopharmaceutical because of the short half-life into Switzerland. So that's where we put up this platform. We put up the radiosynthesis. We collaborate with Avid, with the company, and also with the university hospital. Also, we have set up there also the contracts to be able to manufacture that. And at the end, we now currently still producing the radiopharmaceutical for studies ongoing at the Schibli in Lausanne, but also in Geneva and also in University Hospital in Zurich. So that's really at the point where we have at the moment up to more than 100 patients have been scanned. We have produced, I think this is even outdated, probably more than 100 batches of this radiopharmaceutical. And we have clinical studies ongoing, which are also, we can actually also look the progress in on the web. And you can see here the uptake of the radiotracer in patients. Here, that's an overlay between an MR and the radioactivity. So the green and red spots are the uptake of the radiotracers in Alzheimer traces where you have these tau depositions quite nicely localized, and that's a three-dimensional picture. So this is really to follow those patients over a long period of time. So these studies are lasting now for 2 to 3 years, and they still actually are ongoing for another 2 to 3 years. So that's also a long-term commitment we are engaging into with this platform. Another project, which actually is really bench to bedside from our own research, it relates to folic acid receptor targeting. The background here is that the folic acid receptor targeting drugs for therapy, classical therapy, have failed in the past. Mainly, we think because of a poor selection of the patients. So we have a targeted chemotherapeutic which failed. We have antibodies which have failed, and this is mainly, I think, if you talk to clinicians, could be related to poor selection of patients. So what we did in the past, and this was long before actually the platform was established, we developed such folic acid derivatives. You see here in black the structure of folic acid. We did a number of iterations till we actually reached this compound here, which we called Azafol, which now has been translated to the GMP facility. So we did that again in a multi-center clinical trial. So this was really academic driven. So we developed that, and clinicians, were attracted by the preclinical data. So we have the clinical studies ongoing with the University Hospital Schuf, St. Gallen, in patients suffering from lung cancer, but also ovarian cancer. So this was really a first-in-man study initiated completely into an academic environment. So that's the compound here, and also here the study is currently finished. We could show actually a correlation between folate receptor positive patients, where you see the uptake of the radiotracer in the lesion, and the post-mortem or the immunohistochemistry showing in brown the uptake or the expression of the target, whereas in patients where the receptor is absent, as here, for example, in this lower picture, you could nicely see that this the liver uptake is unspecific. Actually, you see here the black spot, which represents the metastasis, and this is receptor negative, and you have basically here a black spot. The same here in ovarian cancer. Again, here lymph node metastasis, which were receptor positive, also showed up in the patient. So that really is a tool now to establish, to have a selection of patients which are likely to respond on therapy in a classical sense. We are also currently also developing radiotherapeutics for radiotherapeutics for indications in those patients. This is ongoing work, preclinical, but soon also will be translated into a clinical setting. Speaking about translation, this was of course, this is a publication now, in academic publication about this study, which I just presented, and this attracted again the interest of pharmaceutical companies. So this compound has now been out-licensed to Merck, respectively to ITM company in Germany, which is developing radiotherapeutics and need this tracer now for preclinical or for clinical selection of patients. So that's also IP can be generated, and this of course is also be via licenses will be an opportunity to get some funding back. The Denode project, which I'm going to show here, is a therapeutic application of radiotherapeutic development on this platform. Collaboration between the Paul Scherrer Institute and the University Hospital in Basel, where you have a peptide which is targeting the CCK2, the cholecystokinin receptor 2 receptor, which is expressed on medullary thyroid cancer. So these are the thyroid cancers which cannot be treated with iodine, for example, radioactive iodine anymore. Also here we did a proof of concept study, really going from bench to bedside. This was funded by the Swiss Cancer League at this point. For the first-in-man, we did studies, successful studies in patients suffering from medullary thyroid cancer, and these patients do not have any other therapeutic option. So this is really a radiopharmaceutical, which is now being labeled straight away with a therapeutic isotope, namely here in with lutetium-177. So this again, attracted interest of pharmaceutical companies. So Debiopharm licensed this compound and pursued further clinical studies, not only in Switzerland but also outside. And these are other projects which are in the pipeline. I just not going to go into details. So I mentioned Alzheimer's. There is another tracer which has been developed at the ETH and now is going to be transferred in a clinical setting. Also for other indications, for drug development, for example, this can be also used. This is a compound here on the right side on top, which is targeting the glutamatergic system. We have a project which is related to ALS, so amyotrophic lateral sclerosis patient. This is a study which has been launched with the Kantonsspital in St. Gallen, where they have a large cohort of patients suffering from this disease. We have here developed a compound, and it's now going to be produced in the next couple of weeks here at the ETH. Patients will be transferred to the Inselspital in Bern, and this study will be then conducted in Bern with the tracer produced on this platform in those patients, targeting the cannabinoid receptor subtype 2. This receptor seems to be has an implication in neuroinflammation. And of course, that could be a readout, or could be a tracer selecting patients or looking for receptor expression and target expression in those ALS patients. We also have a project with the Kantonsspital in Baden, where they wanted to look into endometriosis. And here is a compound which was not developed in our facility or in our laboratories, but this was adopted from the United States. So again, physicians have seen this tracer and selected as a potential tracer for their indication in Switzerland. It was not available in Switzerland, so we produced this on our platform for these clinical studies. Maybe a few words about costs of such a study and the timelines. We have these numbers are without the clinical service, which it's always involved. Of course, we need also clinicians, which has to, be paid for that. So we're talking about, if it's a first in man, I would say, if really tox is available, the precursor is available, about 100,000 to actually establish such a tracer on the platform. If anything else is already available, tox data, for example, from companies already there, clinical data, dosimetry is available. That is maybe a bit cheaper. But of course, then also setting up these tracers and keeping that in the lifespan of a study, of course, is something which costs. We have personnel. These are clinical studies. We have not a high throughput. So we're talking about maybe 20, 30 patients at the beginning. And so that also, and that over a certain period of time. So we can roughly say 200, 250,000 from our side till a full study can be started in a first injection. So this is a substantial amount of investment. Nevertheless, I think it's still a small number compared to any other drug which would be tested in clinics. Let me just quickly talk about this solid target station and what other isotopes we produce, or exotic isotope we produce. Scandium-44 is such an isotope we would like to produce and are start to produce now on the platform. So what scandium has has decay properties which are suitable for medical application. It's a beta plus particle emitter. It has a half-life of 4 hours, which is more suited than, for example, the short lived which are currently available, and it can be produced via on the cyclotron via this PN reaction. That's a bit of targetry which is needed. So this white spot is actually just the tiny amount of material which is needed to irradiate and to produce the isotope. I'm not going to go into details. It needs also a bit of engineering, and it needs also some chemistry to separate the material. So it's calcium, that's a starting material. When scandium has to be separated, we have to set up such a device to be installed into this platform, that a separation takes place automatically or semi-automated, like over here, because of the radiation, of course, from the material and the process. Nevertheless, this is an optimized process. In 20 minutes, we get the compound. So what is scandium-44 useful for? We have actually produced the isotope in an earlier study together with colleagues in Germany, clinical colleagues in Germany. This has been produced at the platform, shipped to Germany. There it is going to be used for the labeling of a peptide, which is targeting the somatostatin receptor, which is overexpressed in neuroendocrine tumors. So what you see is a patient here, that's the excretion of the radioactive peptide through the kidney. So this is normal excretion, that's the bladder. And you can appreciate those spots of liver metastasis in this patient. So this is not something which is new. I mean, we have other isotopes, other radiopharmaceutical, this peptides labeled with another radionuclide, which show the similar effects or the similar images. However, you know, currently this radiopharmaceutical is labeled with gallium-68, which has a half-life of 1 hour only. So it's you know, the images have to be taken very early after injection, and that's as the images from a patient which has been injected with the scandium-44 peptide over time. And you see after approximately 1 hour where the gallium scan is done, you see the main lesions, but you see the metastasis quite faintly only. So it's because of the tumor to background, so there's still some radioactivity in the background. So with a longer lived half-life where you can actually take scans at later time point, after 3 hours, 4 hours, you clearly can see now, of course, still the main lesion, but you can also appreciate small lesions, which are now much better visible. So a longer lived isotope produced on the platform could also mean a better indication, a better imaging quality and assessment of remaining disease after therapy, for example, whereas with a short-lived isotope currently available, this would not be possible. You miss some lesions. Another aspect I would like to point out is because we just submitted another proposal within the PHRT last call a couple of weeks ago. PET isotopes, so these positron emitting isotopes, they have the peculiarity of these isotopes is that they always decay by annihilation, producing 2 gamma rays of 511 kilo electron volts, which are emitted in opposite direction. So this is a ring detector. So this would be the decay. That would be the gamma rays. And all PET isotopes, fluor-18, scandium-44, all of them, they decay by this mode, and they also primarily emit these 2 gamma rays. However, exotic isotopes like, for example, scandium-44, but also gallium-68 and other exotic radionuclides, which we would like to produce with this solid targetization, they also co-emit sometime to a certain degree also other gamma rays. So you could all of a sudden distinguish an isotope A from an isotope B simply by their co-emission of these gamma rays. Sounds complicated. It is also complicated, and actually is something for the medical physicists to further develop. I just show you here a mixture or a picture of a situation where you have fluor-18, which is a PET radionuclide. We have sodium-22, which is also emitting PET particles or beta particles, but it also co-emits, compared to fluor-18, also co-emits an additional gamma ray. So this red line here, which is 1200 kilo electron volt. So this makes the 2 isotopes distinguishable. With a certain setting, you can appreciate that in this, this is all in fluor-18, this is sodium-22. In field phantom, you can see them fluor-18 on this channel, on this recording, whereas you see the other isotope in another setting of the scanner. And that means that all of a sudden we can actually have 2 information from 2 different traces at the same time. And so that's what we actually want to do. It's this dual isotope PET. And why is this important? And again, this is a clinical, motivation. We have here 2 cases of patients which are suffering from prostate cancer. We have here a the top row, a patient which is performed a gallium-68 PSMA scan. So this is a radiopharmaceutical targeting the prostate-specific membrane antigen. You can appreciate the lesions in this patient. And this is now the same patient scanned with fluorodeoxyglucose, which is a standard tracer used in oncology. And you can appreciate that there is no uptake of FDG except the excretion organs in this patient, whereas you see nicely the lesions with the PSMA. This patient has been subjected to a therapy, a radionuclide therapy with lutetium-177, and you see that the disappearance, you see here the disappearance of all these lesions in this patient. So the therapy is effective. However, a second patient where you have again PSMA scan, you see these lesions here lighting up in this patient. Following up with a fluorodeoxyglucose scan, and you can appreciate there is a mismatch. You see lesions which do only show up with fluorodeoxyglucose in this case, but have only a faint uptake within PSMA. And these patients, after subsequent therapy with lutetium-177, they see progressive disease. So that means this patient, the top patient and the lower row patient, have to be treated differently and have to be assessed differently. However, this is now 2 scans, twice a tracer, twice a CT scan, which has not to be done. That means the patient has to come twice. It has addition dose which comes from the CT, which is necessary in both cases when you use a tracer. So that means it would be much better to have everything at with a one-stop shop situation. That's what we proposed here, is indeed that we are now able, because we have access to isotopes like scandium-44 and fluor-18 on the platform, that we can produce 2 tracers which are simultaneously or almost sequentially in the same session injected into a patient, and by means of distinguishing these 2 isotopes on the scanner, because of their decay properties, you can actually analyze the patient or analyze the outcome of this uptake of the traces in one session. So that means reduce dose to the patient, immediate information for the clinicians which know how to treat those patients, and of course also a better outcome at the end. So that is something which we are now trying to establish. So that means, of course, we have to somehow first produce the tracers, make them available. Secondly, we also have to find algorithm and computer power to establish that. And last but not least, I think time is running. Radiopharmaceuticals is really, that's a time-consuming, a time-critical production. You need to produce them every morning, so that means it's really an early morning. People have to get up to start their cyclotron, to produce the radiopharmaceutical, do the quality control, ship it to the patients, and so that is something which is prone to fail. Every once in a while, you have people doing manipulation, particularly also they have to get up early, so it's not a very attractive working environment. So you have turnover of personnel, and so we together with a company, Augmenticon, and also in collaboration with the Fachhochschule Nordwestschweiz, we launched the InnoSwiss project to actually improve the production of radiopharmaceuticals by implementing augmented reality. And so this is basically that we can train earlier, quicker people to produce radiopharmaceuticals, that there is less failure of production, because patients are waiting in the hospitals. You order your radio tracer, you want to be sure that it's delivered, and if it's not, if there's a failure in production, basically that patient has to be sent home, because there is no alternative to get the tracer in a short period of time. So it's really important also from a pharma company to have a very reliable chain of production. So we try to implement this now with the InnoSwiss proposal, that people can that this production and setting up of the production is supervised via augmented reality. That everything is in place. Needs, of course, software solution, but also technical solutions how to actually operate that. And I hope this video works. Okay, now this is now a dry example how it is performed at the Fachhochschule. They have experts in that. So it's about now taking the right syringe, which has to be prepared, try to get the right buffer system, and all this then should be really optimized, also be checked automatically that even a person which is less experienced is able to produce this radiopharmaceuticals. It's also about, how much of this buffer has to be taken, etc. So these are the first steps with automation of this production. So I think I'm going to come now to the end of the presentation. I think the challenges for the platform is indeed that nevertheless there's high costs of maintenance of the clean room. So we have costs which are higher than maybe in a classical academic laboratory. We need certified equipment. We need trained personnel. Nevertheless, I think the tracer production has to be affordable, so it should not be prohibitive for the clinicians to use new radiopharmaceutical. Sometimes the translation between, or the institutional translation and collaboration is challenging. So we have to deal with legal aspects, hospitals versus ETH, GMP facility. Sometimes the ETH domain cannot adhere to standards which are asked from pharmaceutical companies. So, they have usually big dossiers which you have to adhere on, and the ETH cannot comply always with such regulation and doesn't want to comply to that. And on the other hand, the pharmaceutical companies are not flexible enough to maybe lower their bars when it comes to production of their compounds. Trained staff, I mentioned, this is not difficult and not easy to do, and particularly also in an academic environment. There are increasing hurdles for production of radiopharmaceuticals, but also generally pharmaceutical, according to good manufacturing practice. We need extensive documentations also from the clinical side. So Swissmedic, BAG, Clinical Trial Center, this adds to costs, but also leads to a delay from submission of documents, which also include the pharmaceutical part, so the radiopharmaceutical part, to actually start launching this study. So this time, which is needed 30, 60 days before after submission to actually start a green light from Swissmedic, plus then all these site visits which are necessary from the clinical trial centers, lead of course to a delay where personnel has to be paid, and that's something which sometimes is a bit cumbersome. So let me come to the conclusion now. I think we have shown that there is interest in such a translational radiopharmaceutical platform for precision medicine in Switzerland, and also in Europe. So we see that more and more also from outside, from Europe, they are approaching us. Companies are approaching us to produce radiopharmaceuticals for studies performed in Switzerland. We have a number of examples I've shown you from bench to bedside, even in a clinic, academic environment is possible. We have at this point more than 300 patients overall of all these projects with diagnosed or treated with the radiotracers coming from this technology platform. We had some delay in installation of the solid target because of Corona, and some this installation we had to do and modification we have to do at the ETH, but we are now have approval of the BAG to produce such isotope. And I think a successful clinical study is, of course, also a selling argument for licensing. So I think that is also a platform, is generating additional value to a product when you are able to produce it or have tested it already in patients. So that is really, I think, an important aspect for this platform. I'm again very grateful for the PHRT to approve that, and I hope also our next call or next proposal will be funded in the next couple of months. With that, I would like to acknowledge many people which have contributed, particularly also our clinical partners and also the funding agencies, and I would like to thank you for your attention.